Hello, biology. I just wanted to make a couple clarifications on this assignment that I might not have made in the class recording because I know some of you watch those to help you with the information, so I wanted to upload this with it as well. So here we did go over this in class. That is on the class recording. Just brief reading over, what the heck we've talked about so far and a brief overview as to what transcription is and what translation is. Transcription, during transcription, an enzyme RNA polymerase comes in, and that's what attaches the bases to our DNA strand to give us this RNA strand that you might see right here. So RNA polymerase does that instead of DNA polymerase. mRNA is just a messenger RNA molecule. That's what goes from the nucleus to the ribosomes whenever we are going to translate our protein. So it's exactly what it sounds like. It's a messenger. It carries the message. And then with these, I did go over these answers in class, so that is on the class recording. And then in number 3, you're just summarizing transcription based on the reading from here and the notes that we have taken in our from our Google slideshow. So that's what number 3 is going to be about. Then we get into translation. So we have our mRNA strand, and then eventually that mRNA strand, or that messenger, is translated in our ribosome. So then you're going to see something called tRNA. Basically, this is what's responsible for making those amino acids in our ribosome. So the cell uses transfer RNA, tRNA, to bring the correct amino acid for each codon in the mRNA. So this carries the message. The tRNA brings in the amino acid, those building blocks of our protein. So 3 nucleotides per codon. Okay, the codon is what codes for those amino acids. Again, we talked about those in class. An anticodon is basically when we translate our RNA strand, our messenger RNA, into our amino acids. And I'll show you that here in the table that we're going to have later on. And I'll show you that here. So we're going to be applying transcription and translation into a real life scenario. Sickle cell anemia versus normal red blood cells. So your red blood cells are normally shaped like a donut. In sickle cell anemia, your blood cells are shaped like a crescent moon. And that's an issue because your blood's responsible for carrying oxygen and nutrients. Well, if your blood is misshaped, you're not going to get as much oxygen and nutrients to places where they need to be. So here we're going to show what happens to somebody if they have sickle cell anemia, what goes on here. So let's look at this really quick. Make sure that my pen's working, because normally it doesn't, of course. So we are going to transcribe and translate this DNA strand. So if I'm looking at CAC, that would be my first codon, my first code. So what matches with C? We have G. What matches with A in RNA is going to be U. And then with C, we're going to have G. Now, we're going to translate our mRNA strand into the correct amino acid. So what's going to pair with G? C. What's going to pair with U? A. What's going to pair with G? C. That is going to be our first amino acid. Now we have GTA, Grand Theft Auto, funny. Here's our next codon. So here we're going to have C, and then what's going to pair with T? It's going to be A. And then in RNA, what pairs with A? It's U. Now I need to translate this to my correct amino acid. So what's going to pair with C? G. What's going to pair with A? U. What's going to pair with U? A. And you're going to do that all the way down. Codon, 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 codon. And you're going to continue that exact same process the entire way down our RNA strand. So that's normal red blood cells. Now with sickle cells, you're going to do the exact same thing, but you might see a slight change in your DNA, which in turn is going to lead to changes in your protein. And then you're going to compare the beginning of the proteins versus the end. What is the difference here that leads to that different trait? What is going to give us the misshapen red blood cells? Okay, so this is going to be important when we talk about mutations later on in this unit. Okay, we're going to talk about what happens when there's a mistake in your DNA and how it could drastically affect the traits that you have. Okay, just by one nucleotide being off, it can alter the entire appearance of your red blood cells just by one nucleotide, one base being incorrect. So that's where we're going to be headed after this. That's why we're talking about transcription and translation before we talk about mutations and what happens when there's a mistake in your DNA. Thanks for watching. I hope this was helpful. And you have a great rest of your day. Let me know if you have any questions before your quiz on Wednesday.